This presentation is part of the pharmacodynamics module, and it will address the question, what is meant by the terms drug efficacy and drug potency? At the end of this presentation, you should be able to define the terms drug efficacy and drug potency and explain the relative roles of efficacy and potency in drug selection. When the relationship between drug dose on the x-axis and drug response on the y-axis is plotted on a base 10 logarithmic scale, the resulting dose-response curve is sigmoidal. Increasing drug doses produce increasing drug effects, but this occurs over a relatively narrow part of the overall dose range. Further increases in drug dose or concentration beyond this range produce little extra effect. The maximum response on the curve is referred to as the Emax, and the dose or concentration producing 50% of the maximum response, Emax over 2, as the ED50 or EC50. The curve flattens at high concentration because all of the available receptors have been activated, and so the magnitude of the biological response cannot increase further. Pharmacologists and prescribers often need to compare the pharmacodynamic effects of different drugs in order to choose those that might be the most suitable for their needs, either in drug development or for the treatment of patients. When the curves for different drugs with similar biological actions are plotted on the same graph, although they may have the same basic sigmoid shape, they may lie in different parts of the graph because of differences in their maximum effects and the dose range over which they are effective. These differences illustrate the pharmacological concepts of efficacy and potency. Efficacy is the term used to describe the extent to which a drug can produce a response when all available receptors or binding sites are occupied. This corresponds to Emax on the log dose response curve. When comparing drugs acting at the same receptor, a full agonist will have the greatest efficacy and can produce the maximum response of which the receptor is capable. In contrast, a partial agonist at the same receptor, by definition, will have a lower efficacy. It can achieve only submaximal responses even when all receptor sites are occupied. Therefore, the dose-response curve for a partial agonist is flatter than that of a full agonist and reaches a lower Emax. Therefore, the partial agonist has a reduced efficacy at this receptor compared to the full agonist. This occurs because the molecular configuration produced when it binds to the receptor is less well suited to achieving signal transduction. The concept of efficacy is not restricted to comparing the effects of drugs that act at the same receptor. The term therapeutic efficacy is used to describe the comparison of drugs that produce the same therapeutic effects on a biological system, but do so via different pharmacological mechanisms. For example, loop diuretics such as furosemide have greater natriuretic efficacy than thiazide diuretics such as hydrochlorothiazide. Both act as diuretics, but have their effects at different sites in the renal tubule. The loop diuretic has a higher Emax and therefore greater efficacy, although it requires a higher dose to produce that effect. The treatment of peptic ulcer disease provides another example of the comparative therapeutic efficacy of drugs that achieve the same biological effect but act at different targets. Proton pump inhibitors such as omeprazole are more efficacious at suppressing gastric acid secretion than H2 antagonists such as cimetidine. Potency is a term used to describe the amount of drug required to produce a given response. More potent drugs produce biological effects at lower doses or concentrations so that they have a lower ED50. The potency of a drug is related to its affinity for the receptor, which indicates how readily the drug-receptor complex is formed. In this example, drug C is the most potent because drug responses are achieved at 10-fold lower doses than drug A and 100-fold lower doses than drug B. It is important to note that a less potent drug can have an efficacy similar to or greater than that of a more potent one. In this example, drug A is less potent than drug C, but is more efficacious. Differences in potency can be readily overcome by giving less potent drugs in higher doses. Although the efficacy of several individual drugs in the same drug class may be broadly equivalent, their potency can differ considerably, and this is reflected in the different recommended dose ranges. For example, the recommended dose ranges of H2 antagonists used to treat peptic ulcer disease and ACE inhibitors used to treat cardiovascular disease differ considerably, although there are no differences in the efficacy of the drugs within each drug class. Efficacy and potency are clearly of relevance in the development of new drugs. Candidate molecules that have no efficacy or have very low potency are unlikely to be useful. However, of those drugs that have been licensed for use, relative potency or efficacy is rarely a consideration when considering whether one drug is preferable to another. When judging the relative merits of drugs for a patient, prescribers should consider other important factors such as the overall adverse effect profile, the therapeutic index, the ease of administration for the patient, the duration of the effect, i.e. the number of doses needed each day, and its cost. So do potency and efficacy ever matter in drug selection? Differences in relative potency can usefully be overcome by altered dosage. However, it should be remembered that most of the adverse effects of drugs are also dose-related. If the adverse effects occur by a mechanism other than the receptor-ligand interaction that mediates the beneficial effect, then potency may be relevant. This is because only the more potent drug will produce beneficial effects at the low concentrations that can avoid unwanted adverse effects. It might seem logical to choose a drug with the greatest therapeutic efficacy when looking to select a drug with beneficial effects from a group of similar drugs. However, in some cases the most efficacious drug may be less preferable because the mechanism of action that leads to the clinical benefits may also be responsible for causing dose-limiting adverse effects. For example, beta-1 adrenoceptor blocking drugs exert their anti-anginal effects at the heart by blocking the actions of catecholamines at beta-1 adrenoceptors. This pharmacological action is also responsible for causing important dose-limiting adverse effects such as bradycardia and heart block in some patients. Drugs in this class with the greatest therapeutic efficacy would also be more likely to cause these adverse effects. When the same action leads to both the beneficial and adverse effects, the latter can be minimized by carefully titrating the dose. However, some drugs have a steeper dose response curve, which makes them more difficult to titrate to the dose that is effective but that avoids adverse effects. In these circumstances, it's going to be preferable to choose the drug that prescribers can most safely titrate rather than the one that is most efficacious. That brings me to a brief review of the key points of this presentation. The efficacy of a drug is a measure of its capacity to produce a biological effect. The maximum biological effect produced by a drug is known as Emax. The term therapeutic efficacy is used to describe the comparison of drugs that produce the same therapeutic effects on a biological system, but do so by different pharmacological mechanisms. The potency of a drug is an expression of the amount of a drug required to produce biological effects and is normally expressed as the ED50 or the dose required to produce 50% of the maximum effect. Efficacy and potency are rarely important factors in drug selection. You can find further reading and resources to support your learning at the locations shown. I hope you found this presentation helpful and look forward to you joining me again for more clinical pharmacology at this site shortly.